All right, so this is the second lecture of this third unit, which is the spinal cord. So we did nervous tissue last time, and now we're going to look at the two organs of the central nervous system, which are the spinal cord first, and then we'll look at the brain after that. So we'll start off by looking at some just general anatomy and get a sort of architectural understanding of how this spinal cord is set up and organized, and then we'll look at some physiological concepts, primarily look at reflexes and how those are passed through the spinal cord and sort of how those work. So as I said, spinal reflexes are what the primary thing that we'll be looking at as we go through our lecture. And these, of course, are, as you guys know, rapid automatic responses triggered by some sort of specific stimuli, and they're controlled by the spinal cord. The brain is not sort of getting involved at this time. This, for speed purposes, right? If you touch something that's hot or you're in a dangerous situation, you need a reflex to remove yourself as quickly as possible. You sort of want that reflex to happen very fast, and so the fact that it can just go to the spinal cord and back to the muscle to cause a contraction is faster than taking the time to go up to the brain and then coming back down. So they're rapid, fast, really quick automatic responses that become built in. And so we'll look at spinal reflexes and reflex arc and sort of see how that works. There are cranial reflexes as well off of cranial nerves. We'll talk about those next time we look at the brain. All right, so anatomy of the spinal cord. It's about 18 inches long. Basically starts obviously at the end of the medulla oblongata, which is the last part of the brain stem, and then runs the length of the spinal cord, truly ends around L1 or L2, and then from that point it starts to taper a little bit and sort of take on a different form. But it's about a ½ inch wide and has what we call bilateral symmetry, which is basically giving a left and right. These grooves is posterior median sulcus and anterior median fissure, which are grooves that divide it in half, essentially, into left and right. And your brain is the same thing. You're going to have two separate, distinct hemispheres, but there are certain locations where information can cross over. Same with the brain, same with the spinal cord. The brain, one of the primary areas for that is called the corpus callosum. In the spinal cord, it's very close to the central sulcus, or the central canal of the spinal cord, and so we'll see how that works coming up. All right. So the spinal cord also has enlargements, which are caused by an increase in gray matter in these regions. And there's a cervical enlargement and a lumbar enlargement. And basically that is because there are increased amount of nerves dedicated for the shoulders and the limbs. And the same thing in the lumbar enlargement. You have more nerves needed for the pelvis and the lower limbs. And so you're actually getting enlargement of that area. Okay? The distal end of the spinal cord, as I said, sort of changes below the lumbar enlargement into this thing called the filum terminale, which is, sort of a thin thread of fibrous tissue, and ultimately it turns into what's called the cauda equina below that conus medullaris. As you can see in this image, you can see the cervical enlargement and the lumbar enlargement, and then again you can see at the conus medullaris, it really starts to change and taper into what we call cauda equina, which means horse's tail, and it spreads out and sort of fans out in that fashion as it goes down into the sacral spinal nerves. Right? And then again, if you look at a cross-section of the spinal cord, again, you can see these divisions, right? You have an anterior median fissure here, right? And then you have a posterior median sulcus here, and giving you sort of a left and a right side to the spinal cord. You can also appreciate gray and white matter. We talked about gray matter and white matter before. Gray matter is primarily neuron cell bodies, whereas white matter is primarily axons. And so that's what you're seeing in terms of how that is organized. Okay? There are 31 spinal cord segments based on where the vertebrae and the spinal nerves originate. The cervical nerves are named for the inferior vertebrae. All the other names are named for the superior vertebrae. And you see these spinal nerves. And so you're learning that in lab right now. When you look at the vertebral canal and you see as the vertebrae line up with one another, it creates what are called intervertebral foramen as these adjoining vertebrae come together, allowing for openings for these nerves to exit from the spinal cord. All right, so the spinal cord is divided into two branches that we call roots. And if you look at the image, you can see there's a ventral root and a dorsal root. And basically, the dorsal root contains the axons of sensory neurons, so information coming in, and then the ventral root has axons for motor neurons going back out. And so it's sort of divided up, and then ultimately that extends out and forms what's called a spinal nerve. The dorsal and ventral roots join, form a spinal nerve, and then ultimately they're able to carry both sensory and motor information. But as it gets very close to the spinal cord, you see the split, and into this sensory information coming in, motor information going out. And so you can sort of see that right here. Here is the dorsal root, right? So information coming into the spinal cord goes this posterior route into the spinal cord, and then motor information coming out will then hook up with a neuron and come out this ventral root. And then again, as you see, it joins into this common spinal nerve, and then we'll head out to the target. Okay, so sensory information comes in the dorsal root, and a corresponding motor response then goes out the ventral root. And that's what's called a reflex arc, and we'll look at reflex arcs in a little bit coming up. Also in this image, you can see the meninges. Right, the meninges are a three-layered membranous system that is designed to isolate and protect the spinal cord, and is also actually continuous in the brain. So you have a spinal meninges and a cranial meninges, and they're basically made of the same materials, and they're designed really to protect the brain. And so here's a nice image, again, of a cross-section through the vertebrae and the spinal cord, where you can see the meninges, and you can see the bone of the vertebrae and those components there. So the spinal meninges, as I said, is a specialized membrane system to isolate the spinal cord from the surroundings. Remember, the central nervous system, the brain and the spinal cord, very delicate tissue that cannot survive in the, just general interstitial fluid. It would be far too toxic for this nervous tissue, and so it needs to be isolated. And there are many different ways we isolate this nervous tissue. We can isolate it biochemically, we can isolate it physically, and this is one of the ways we physically isolate it. This membranous wrapping system protects the spinal cord. It's continuous, as I said, with the cranial meninges, but also as a route for blood supply into this region. And so you've probably heard of meningitis. Meningitis is just sort of an infection or inflammation of this meningeal system. Either a viral or bacterial infection can occur, and can be very dangerous, right? And part of the reason for that is that it's continuous with the cranial meninges, and so very easy for infection to work its way up to the brain, which obviously can be very detrimental. So if we look at this spinal meninges, and again with the cranial meninges, you're both components, you're going to see three layers. You're going to see a dura mater, arachnoid mater, and a pia mater. Okay? Dura mater is the outermost layer. It's a pretty thick, tough, fibrous layer. You'll have the opportunity to see this dura mater when you do your brain dissection coming up later in the semester, and that's the first thing to pull off the brain is this thick, tough, fibrous dura mater on the outside. Cranially, it actually fuses with the occipital bone and caudally tapers to form the coccygeal ligament, and so essentially this not only is it providing a nice, strong outer protection, but it's also anchoring the nervous tissue inside those components. So the dura mater also has what's called an epidural space. Epi meaning above, dura meaning the dura mater. So this is the space between the spine, the dura mater, and the walls of the vertebral canal. And in this location, if you look at the previous image, you can see adipose tissue is found in this, so loose connective and adipose tissue is found. And you may have heard of an epidural or an epidural injection before. Like if you've given birth and maybe had this done to yourself or know someone that has, right? This is how an anesthesiologist delivers an anesthetic injection to a woman who's about to give birth, right? They want to sort of numb the area, and the injection site is delivered directly above the dura mater. So the needle's injected or inserted in between the vertebrae and basically goes into that adipose tissue area, and that's where the anesthetic is released into that area and numbs up the area. Next layer is what's called the arachnoid mater. So this is the middle meningeal layer. The arachnoid membrane has a simple squamous epithelia that covers that arachnoid mater, and a couple of components of it. You have a subdural space between the arachnoid mater and the dura mater, but you also have what's called a subarachnoid space, which is, of course, deeper between the arachnoid and then the pia mater. And here, this is very important because this is where we have CSF. We talked about CSF being in the central canal of the spinal cord and in the ventricles of the brain, but also moves out into the subarachnoid space. So not only is it inside the nervous tissue, it's also surrounding the nervous tissue. And so in this subarachnoid space, CSF can be withdrawn, right? And you may have heard of a spinal tap or a lumbar puncture. That needle, again, similarly to how we talked about the anesthetic injector, will be inserted. This time it's going to go through the dura mater, through the arachnoid mater, into the subarachnoid space, and then just like drawing blood or something, you can pull the fluid, withdraw the fluid into a syringe, and then analyze it, because the CSF is going to carry dissolved gases and nutrients and waste products and proteins and other types of things that may give you an insight into what is going on with someone's brain, right? If they're having issues or suspect Parkinson's disease or Alzheimer's or something, the physician might do a spinal tap to sort of assess of what's happening there. So that's the CSF in that subarachnoid space. Also in that subarachnoid space, again, you're going to see vessels, blood vessels, and unfortunately one thing that can happen, a type of stroke that can occur, is called a subarachnoid hemorrhage, something you may have heard of, where you actually get bleeding in that subarachnoid space. And so that can be very problematic because obviously you have fluid in that area already with the CSF, but with additional bleeding that's occurring there, you can try to drain that additional blood and fluid that's coming in that area to how to protect the brain from further injury. All right, so the innermost layer is the pia mater. And this is basically sort of a thin mesh of collagen and elastic fibers that's actually directly bound to the neural tissue beneath it, right? So it's when you do your dissection of the brain, you'll pull off the dura and the arachnoid, and you'll be able to see just sort of a glossy membranous covering, and that's the pia mater, covering that nervous tissue. Okay? And so, again, blood vessels will be found in this area through the subarachnoid space onto the surface of the spinal pia mater, and you can see in this image, you can sort of see that glossy covering that I was talking about. They sort of peeled away a little bit of it on the spinal cord. And you can see the anterior median fissure here. You can see the dorsal and ventral roots over here. The dura mater is sort of pulled back a little bit there. You can see that. And you can also see a nice blood vessel in that location. So a lot to see there in that image of the spinal cord. Now, as I said, the sexual anatomy of the spinal cord allows you to see the white matter and the gray matter. The gray matter is, again, containing primarily neuron cell bodies and glial cells, and we call these area gray horns, right? And the white matter is organized as what we call columns, essentially, posterior, anterior, and lateral white columns. So if you look at this image, this gives you a really great understanding of how this system is organized, in that you have the dorsal root over here, sensory information coming in, right? It's going to hook up with a motor neuron here and then go back out the ventral root back out to the target. Okay? And so that, again, that's what we call a reflex arc. But you can also see easily how this gray matter and white matter are organized, right? The gray matter is organized in what we call horns, posterior, anterior, and lateral horns. And then the gray matter is organized into what we call columns, right? So you have white column, lateral white column, anterior white commissure, and you can see those areas where information can cross over, right? Because here you have your posterior median sulcus, here's your anterior median fissure. We have this little space here. So if information does need to cross over to the other side, that's where that's going to happen. Okay? So just make sure you have a good understanding of how the spinal cord is organized with the gray matter and the white matter, and have a really good understanding also of the dorsal pathway and the ventral pathway, and that means sensory information coming in, motor information going out. And there's another actual micrograph of the spinal cord cross-section. All right, so this sort of summarizes a lot of things we talked about, so if you want to look at that to review, you can. But now we want to move on to reflexes, which again is sort of the primary physiological concept that we want to take a look at and sort of see how these work. And reflexes are basically automatic, coordinated responses within the spinal cord, and they will involve a sensory neuron, a motor neuron, okay, and then sometimes will also involve interneurons. As I talked about before, interneurons allow for more complex reflexes, but you may have sensory or motor neurons that would allow for more simpler reflex. So, neuroreflexes again are rapid automatic responses to some sort of specific stimuli, and these are the basic building blocks of neural function. So oftentimes, you go for a physical, a physician's going to do a reflex at your knee, the knee jerk reflex, we're all familiar with that. When a newborn comes into the world, one of the things a pediatrician does is test for certain reflexes to make sure those are present, because again, it demonstrates normal, healthy neural function. Without those reflexes, they may be indicative of some issue or some problem. So one neural reflex typically produces one motor response, and the wiring of this reflex pathway is again what we call a reflex arc. It begins at a receptor, ends at an effector, and typically is sort of a negative feedback to oppose that original stimulus. Okay? So what we want to do is take a look at the 5 steps involved in the reflex arc, and so this is something you want to make sure you have a good understanding of in these 5 steps. Okay, and so the first thing that happens, and this visual shows basically the same thing. First thing is you have arrival of the stimulus, therefore activating the receptor. So in this case, the person's hand looks like it's been stimulated by a tack or a nail or something like that. Right? It stimulates the nerve ending, and then therefore activates the sensory neuron. Sensory neuron sends an impulse back to the spinal cord, of course via the dorsal root. Right? Comes in through the gray. Posterior gray horn. Information processing then occurs in the central nervous system, right? And so in information processing, that could be an interneuron that could then relay that information up to the brain, for instance, but also could send an impulse to a motor neuron, which would then send an impulse out the ventral root to the skeletal muscle. So step 4 is activation of that motor neuron. And then, of course, the result will be response by the effector. So 5 steps involved in this reflex arc. Activation of the stimulus, arrival of the stimulus, activation of the sensory receptor, information processing in the central nervous system, activation of the motor neuron, and then finally response by the effector. All right, and that pulls the hand away, right? So quick response causing the muscle to contract. Okay? And so reflexes can be classified by development, type of motor response, how complex they are, and then also by information processing. So developmental reflexes, right? Some are innate, some things we're born with, right? They're actually formed before birth. Those are the type of reflexes I said, like a pediatrician will come to the hospital first day after a newborn arrives and test a lot of these reflexes, and they're supposed to see certain things. But there are some that are acquired, right? Rapid, automatic, learned motor patterns that we learn, like in, playing a sport, for instance. If someone plays a sport, they're going to learn certain reflexes that maybe someone who didn't play that sport may not have those reflexes, right? They're acquired, they're learned. But they do become rapid, sort of automatic responses. There also are the type of motor response. Of course, somatic reflexes, right, are reflexes of the skeletal muscular system typically, right? So like the knee jerk reflex is a somatic reflex. But there also are visceral reflexes. We'll talk a lot about autonomic reflexes, things that control other than the muscular system, like in digestion or urination, for instance. We have the micturition reflex in the urinary system that makes us want to urinate, right? And so it's a stretch reflex that stimulates our brain and lets us know that it's time to release the urine from our bladder. And so that's what we would call a visceral reflex, right? So we'll talk more about those in NP2. For now, we're going to focus mostly on somatic reflex. Reflexes can also vary in complexity. I talked about this previously. You can have monosynaptic reflexes, which are, where you have one specific sensory neuron linking up with a motor neuron. Nothing else, just one synapse between a sensory and a motor neuron. Or you can have more complex reflex responses that require multiple synapses, and we call those polysynapses. So this is something that I want to talk about a little more here in a minute, is monosynaptic and polysynaptic reflexes. And then finally, the side of information processing. As I said, there are cranial reflexes, and those are going to, of course, occurring in the brain. Spinal reflexes are going to occur in the spinal cord. All right, and so this gives you a nice overview of how reflexes can be classified by development, type of response, complexity, and where it's processed. All right, so as I said, I want to spend a little bit more time talking about the different types of spinal reflexes that can range based on complexity. We have very simple reflexes, and we can have more complex reflexes. So monosynaptic reflexes are going to be the more basic ones. Polysynaptic reflexes are going to be more complex. And so monosynaptic reflexes, again, have the least delay between sensory input and motor output when you need something to happen very, very quickly, for instance, right? And so, for example, the stretch reflex, right? Like the patellar reflex, where you're having a physical and you're sitting up and the doctor strikes the knee tendon with a little mallet and your knee kicks up, right? We've all experienced that before. And this happens very quickly, 20 to 40 milliseconds. And that's a great example of a monosynaptic reflex, right? You have the person's leg sitting down, you're putting a stretch in the muscle, the stimulus is applied to the patellar tendon, that activates the receptor in the muscle spindle. That sends an impulse up to the spinal cord via the dorsal root, of course. So as you can see, there's just one sensory neuron and there's just one motor neuron. So there's only one synapse, hence the term monosynaptic. That then hooks up with a motor neuron, sends it out to the muscle. The effector results in contraction of the leg, and that ends the reflex. So that's a great example. Stretch reflex is a great example of a monosynaptic reflex arc. Okay? And so this image just shows where the receptor is actually in the muscle fiber itself, and then ultimately the muscle will respond by contracting. So that's a great example of a monosynaptic reflex. Polysynaptic reflexes are more complicated, right? They have typically interneurons. Remember we talked about interneurons being a type of neuron that stays just within the central nervous system to control more than one muscle group. Typically they're going to produce either an excitatory response or an inhibitory response to allow for a more complex response. So a withdrawal reflex, for instance, is a great example of a polysynaptic spinal reflex, right? To move your body away from some sort of stimulus, like if you touch something that's hot or sharp or whatever, right? That is involving a polysynaptic reflex. So as you see in this example, the flexor reflex, you have a painful stimulus, of course, that activates the sensory receptor, sends an impulse up to the spinal cord via the dorsal root, of course, hooks up with an interneuron that may send information up to the brain or other locations in the body, but then also will hook up with a motor neuron to stimulate the flexor to contract and pull your hand away, right? But you may also see inhibition of the extensors to allow that muscle to contract. And we'll talk about that a little bit more in a minute. But as you can see, there are 1, 2, 3, 4 synapses here. So, right? So poly, right? More than one. So that's a great example of a polysynaptic reflex. But again, the difference is this will take a little bit longer, right? Because there's synaptic delay, and so each time the depolarization and release of the neurotransmitter occurs at these components, you have to account for that time. So it's a little, just slightly longer than a monosynaptic reflex. So, as I said before, this concept known as what we call reciprocal inhibition is when you get inhibition of an opposing muscle group to allow for this flexor reflex to work. So the stretch reflex of the extensor muscle must be inhibited by interneurons of the spinal cord. And so that's what we saw with the tricep, for instance, being inhibited, allowing the bicep to pull the hand away to allow for prevent competition between the two muscle groups. All right, so reflex arts typically are what we call ipsilateral, meaning they occur on the same side of the body as the stimulus, right? You touch something on your left hand that's hot, you're not going to pull your right hand away. Typically, the left hand then will move away, right? And so stretch, tendon, withdrawal reflexes are all great examples of ipsilateral reflex arc, okay? But there are occasions where we need what we call a contralateral reflex arc, where it actually crosses over to the other side of the body, okay? And a cross-extensor reflex typically occurs simultaneously, coordinated with that flexor reflex, okay? And so a great example of that cross-extensor reflex is like, for instance, when we step on something that, is sharp, right? I talked about this previously, but, if you step on some sort of painful stimulus, obviously you're going to get a withdrawal reflex to pull your leg up from that stimulus, right? Just like we saw when you pulled your hand away from the hot stove, okay? But in this case, you also need the impulse to cross over to the other side of the body, right? So you don't fall over. So you get the extensor stimulated, the flexors inhibited, and that sort of allows you to, maintain balance. And so as you can see, again, many synapses, if you look up at the spinal cord, 1, 2, 3, 4, 5, 6 synapses here. So again, great example of a polysynaptic reflex that also crosses over to the other side of the body. So we call it the crossed extensor reflex. All right. So to wrap things up, I thought clinically we talk a little bit about what's called amyotrophic lateral sclerosis, or ALS. ALS involves these actual motor neurons that we've been talking about, and ALS is a lethal, incurable neurodegenerative disease that basically degenerates these motor neurons, which leads ultimately to muscle atrophy. And then typically the sort of fatal event is respiratory muscles aren't able to work because of the lack of the impulse, and so they atrophy so significantly that respiration is not possible. So most of the cases of ALS is unknown. Okay, 90% of these cases are sporadic, meaning they have no genetic linkage. 10% are linked to inherited mutations, and of that 10%, 2% are caused by mutations to a gene SOD1 or 31. They oftentimes SOD1 is studied quite a bit to try to give us clues to perhaps understanding what's happening in the 90% of these sporadic cases. But still, at this point, there isn't a lot of progress made on ALS. And so, as you can see in this image, these are motor neurons, right? Motor neurons are going, somatic motor neurons are going out to skeletal muscle. In a normal, healthy condition, you have a nerve cell, right? It's sending an impulse down a myelinated axon, and it's going to move, and the muscle is going to contract. And when the muscle contracts, right, it stays very healthy. We talked about this with skeletal muscle. Use it or lose it, right? If you don't contract a muscle, it will atrophy, right? And so in ALS, the motor neurons are destroyed and damaged. The impulses don't get to the muscle. Result is a wasted muscle, and the muscle atrophies and cannot contract, okay? Among these sporadic cases, most widely held theories are issues with the glial cells, excitotoxicity, or perhaps exposure to some sort of metal or pesticide that could potentially cause these reasons. The other 10%, again, are specifically related to mutations in genes. And so one of those, again, is the superoxide dismutase, SOD1 protein. Misfolding of this protein basically leads to destruction of these neurons that regulate these muscle contractions. And so there are some new treatments out there trying to look at perhaps blocking the production of SOD1, maybe silencing these individual genes. But again, whenever you sort of play with this genetic side of things, you get potential side effects which could cause even further issues. So, but SOD1 is a pretty well-studied pathway in trying to understand the sort of genesis of this disease. All right, so death of motor neurons, again, in ALS leads to difficulty breathing and swallowing, weakness, muscle atrophy, paralysis can occur. Typically, people are going to survive maybe just 1 to 5 years with this. And again, ultimately, failure of the respiratory muscles is the fatal event that occurs. The selectivity of the neurons is also sort of baffling and interesting. There's no cognitive impairments. Neurons to the bladder and the eye are typically spared, so it can be, very challenging from that standpoint. Riluzole is the only medication approved to treat ALS, and you have a very modest effect on average only improves survival by a few months. So typically non-pharmacologic therapies like nutrition and respiratory function, occupational, psychological support, speech therapy, all these types of things are really designed just to help minimize the effect and try to improve the quality of life for these individuals that have this devastating disease. All right, so that's it for the spinal cord. We will wrap it up with the next chapter, which is going to be looking at the brain, of course, and that'll be the last chapter for this unit. And then we'll move on to the last unit after that. So look for the next brain lecture coming up soon.